The Calvin cycle is the second part of photosynthesis. It uses the chemical energy in ATP and NADPH created during the light reactions to reduce carbon dioxide and make sugar molecules called G3P. The sugar can be used to synthesize other organic materials such as carbohydrate, glucose.
Stage 1 of the Calvin cycle is the carbon fixation stage. This begins by the CO2 molecule combining with RUBP, or ribulose bisphosphate, which is a 5-carbon sugar. The enzyme Rubisco allows this to happen by catalyzing the reaction. The combination of CO2 and RUBP creates an unstable intermediate made up of 6 carbons, which splits into 2 molecules of 3-phosphoglycerate.
Stage 2 is the reduction stage, in which ATP phosphorylates 3-phosphoglycerate and NADPH reduces the 1,3-bisphosphoglycerate and gets rid of one phosphate group to form PGAL or G3P. One net G3P is produced in 3 cycles, so in order to gain 2 G3Ps to form glucose, the Calvin cycle must be turned 6 times. Go.
Stage 3 is where RUBP is regenerated so that the Calvin cycle can continue carbon fixation and create more G3P molecules. The inner workings are complex and include a series of reactions, but the net result is 3 RUBP molecules for every 5 G3P molecules. Thus, one of the G3P molecules created exits photosynthesis to be used for further synthesis of organic molecules. The other 5 3-carbon G3P molecules are rearranged into RUBP, and 3 5-carbon molecules. In order to carry out these reactions, 3 ATP molecules must be consumed.
In total, 9 ATP molecules and 6 NADPH molecules are needed to generate 1 molecule of G3P. These molecules are created in the light reactions of photosynthesis. Thus, sugar cannot be created without incorporating both the light reactions and the Calvin cycle. These sugars can then be used in various ways, such as cell respiration in the plant as well as heterotrophic organisms.